Thank you for the lovely introduction, and thank you, Oxford Nanopore, for providing me with the opportunity to present our data here. So no conflict of interest, yes. I think my colleagues have already gone through the scope of the problem of the increasing complexity of diagnostics and the multitude of different tumor types in CNS tumors have made, increased financial burden and lengthened the turnaround times for diagnostics. There are many different markers to look for, as we previously discussed: copy number variants, gene amplifications, homozygous deletions, point mutations, and structural variants. And also methylation classifications, which has been a really big game changer for the whole field. So the current methodology to get these markers is to run them on multiple different platforms, like methylation bead arrays, NGS panels for SNPs, RNA fusion panels to detect gene fusions, and FISH to detect homozygous deletions or focal amplifications. Now, what will typically happen in many of these genomics labs, of course, is they run on a need-to-know basis. So it's decided for every case what the initial panel will be, and if that gives the necessary answers, then you have your diagnosis. But in many cases, you have to continue to the next panel, and then the next panel, and so forth. So the turnaround time can be very long. But all of these markers and assays are uniquely covered by the PromethION sequencing platform. And with the development of the PromethION flow cell, it should be possible, hypothetically, to get all you need from a single whole genome sequencing run. So we set up a study with an emphasis on simplicity. So we would be running one sample per flow cell. We only run tumor samples, no normal samples. We'd get all the information that we can from a single whole genome sequencing run, and we'd compare the diagnostic results from that with that of standard of care from our institutions. We ran 58 samples from a tissue biobank, and finally 20 samples were run prospectively, so that we did the Nanopore analysis, whole genome sequencing analysis, at the same time as they went into standard of care. So in a nutshell, the pipeline, our workflow is that we get the tissue biopsy shortly after surgery. We perform DNA extraction on spin columns. There's no size selection. There's no shearing. We do adapter ligation for library prep, and then the sample is loaded on a flow cell and sequenced for the duration of that flow cell, which is typically 72 hours. Base calling is done live with methylation. Alignment is performed live, so at the end of the sequencing run, you will have all the data to continue to the final analysis. Now, from there, we do copy number variation analysis, methylation classification, structural variant calling, and small variants. And all of the heavy lifting here is done on the P24 tower and epitomy lab. So it's like this is the out-of-box capabilities of the PromethION platform. Simultaneously, the sample has gone through standard histomorphological evaluation. So when the results from these two arms are ready, we are ready for an integrated analysis. So the basic sequencing metrics, we were aiming for 30x whole genome coverage. We performed a little bit better than that. I think the average whole genome coverage in our cohort is around 40x. The read length distribution hovers around 10 kb, so that's just how they come from the spin columns. Tumor cell content is really important here, because obviously we're not going to extreme depths of coverage. We set a hard sort of cutoff of 15% tumor that would probably not be enough to get any relevant information. The majority of our samples were higher than that, and this is evaluated from the copy number variant profiles after the sequencing. We also evaluated tumor cell content from frozen slides prior to sequencing, and we can say that like if the pathologist can detect and see tumor cells from the frozen sections, they're usually good to go. What do the results that we get look like? Now we do methylation-based classification with two different classifiers, the cross-NN classifier that was developed in Charité in Berlin, and the newly released MMP Flex classifier from Heidelberg, which is based on the V12 version of the classifier. We get copy number profiles, for example here where we see a gain of chromosome 7 and loss of chromosome 10, in addition to an EGFR amplification and CDKN2A homozygous deletion like Mashat mentioned in her talk previously. Gene fusion events are detected as structural variants and annotated with SVANA. In this case, we have a breakpoint on the X chromosome and chromosome 22 causing a EP300 B-core fusion, which is pathognomic for a new entity that was first described a few years ago. Single nucleotide and structural variants are called and annotated, and we can pull out screenshots of the specific region. We have a TERT promoter variant on the left and a structural variant in EGFR on the right that causes the deletion of 6 exons. Finally, MGMT promoter methylation is very important in prognostication and drug treatment of glioblastomas. We get a very detailed map of methylation on the MGMT promoter with Nanopore data instead of like 1 or 2 specific sites with the standard of care assays. So how did the cohort perform with relation to standard of care? This is a complicated slide. I'll walk you through everything, but notice all the gray triangles. That means that there was no data from standard of care to compare it to because they run as limited data as they possibly can. So concordance of methylation classifiers, as we discussed previously, we had 88 to 92% concordance with the final diagnosis. Copy number variants, we detect everything that was detected by standard of care with 100% sensitivity. We have 89% precision, and that is because of 4 samples that were standard of care is fish that they reported a negative results where we found small or complex deletions of CDKN2A. So I'd say that even though the precision is a little bit lower, that's because we're detecting more. On single nucleotide variant level, we have 97% sensitivity, 100% specificity. We were calling no false positive. The first 5 there are diagnostically important variants, and those were in 100% concordance with standard of care. We call variants many other genes that are predicted to have some diagnostic relevance or treatment relevance. So those were also, we call it in a lot more samples because we have them. But all in all, I think we have pretty good results with this. Gene fusions, diagnostically relevant gene fusions were detected in 3 samples that was concordant with standard of care. We also call structural variants that are generally not called with standard of care. And MGMT methylation stratification had 94% concordance with standard of care. Finally, the return time, running everything on a single assay like this obviously makes the whole diagnosis process a lot simpler. We were able to return the results within a week in every case. Our median return time was 5 days, which is considerably better than standard of care, although I'd say 9 is probably fairly good for a large institution, but that excludes sort of the samples where you had to go into more complex analysis, which took many weeks, or in some cases months, to return the final classification. So with that, I will thank you for your attention. Leave you with a picture of Vilhelm Magnus performing one of the first open brain surgeries in Oslo in the early 1900s, and hope we can all appreciate how long we've come since then. And I'd like to thank my colleagues and give a special shout out to Professor Einar Vikmo, who is a neurosurgeon and the group leader at Vilhelm Magnus. Very forward-thinking, got us into intraoperative diagnostics very early, and we're continuing on that journey. And Henning Leske at the Department of Pathology, which has been very open to new methods and better treatment options. Thank you.